We're so late getting started. Where's everybody? The back row's empty today.
It's Friday.
Jenny's
so
gone.
Elizabeth has been painting again. So, we said we would start the citric acid cycle today. Are there any mop-up questions on glycogen before we get started? Glycogen made sense? The control of glycogen makes sense?
There were a couple of things that we didn't specifically talk about that were like kind of obvious that I assumed. That one figure that had all the control on it, and it was like glucose and something else that we didn't talk about, but that's kind of just, we're still responsible for it and stuff.
Yes. Oh, yeah. Yes. Those, you mean like the trends of control?
With all the glycogen phosphorylase stuff. I'll look at it later.
I'm confused. That's okay. That's not unusual. Okay? Robbie, nice of you to join us. So we're going to move to the citric acid cycle. Now, we've done a lot of stuff. And again, when we talk about the citric acid cycle in general biology, when we talk about the citric acid cycle in physiology, it tends to come right after talking about carbohydrate metabolism, right? When we did glycolysis, we sort of ended glycolysis with pyruvate. And so we tend to start the discussion of the citric acid cycle with pyruvate, right? But we have to remember, the citric acid cycle is the central sort of metabolic pathway cycle in metabolism. It's involved in all kinds of processes. Not just breakdown in terms of energy metabolism, but also providing raw materials for some other processes. So we've got pyruvate, which could come from carbohydrate. Where else might we get pyruvate? What? Amino acids, right? So some amino acid carbon skeletons end up in pyruvate. Specifically, alanine, right? Alanine is metabolized directly through pyruvate. We talk about the conversion of pyruvate to acetyl-CoA. That's where we will start. But where else can we get acetyl-CoA? Fatty acid metabolism. What else? Robbie? Amino acids, right? Some amino acids are metabolized like lipids, and so their carbon enters at acetyl-CoA. Right. Intermediates in the citric acid cycle are used for other things. We talked about citrate being a control molecule for glycolysis. Citrate is exported from the mitochondria, not just as a control molecule, not just as a signal for glycolysis, but it's also the way that 2 carbon units are carried out of the mitochondrion for fatty acid synthesis. Right? So citrate can be taken out of the citric acid cycle for other pathways. Any amino acid that is metabolized through the amino acid glutamate, right? Glutamic acid. If we deaminate glutamic acid, we get alpha-ketoglutarate. So amino acid carbon can enter and leave the citric acid cycle here at alpha-ketoglutarate. If we're talking aspartate, right, 1 carbon shorter, aspartate can be converted into oxaloacetate and vice versa. So carbon can come in or leave here with, at oxaloacetate during amino acid metabolism. We talked about malate and oxaloacetate being involved in gluconeogenesis, right? That pyruvate was carboxylated to oxaloacetate. Oxaloacetate was converted to malate. Malate was exported from the mitochondrion as part of gluconeogenesis. So the citric acid cycle is central to a number of processes, both catabolic processes, that is breaking down things in order to get smaller molecules and extract usable energy from them, and anabolic processes. That is, the citric acid cycle can provide small molecule precursors for the synthesis of other things. So there's a lot going on with the citric acid cycle, right? It's not just the endpoint of carbohydrate metabolism. There's all kinds of things that the citric acid cycle is involved with. Question, Sarah? You've got a questioning look on your face.
No, that's just like, I was trying to take notes of all the stuff you're saying. That was a lot of information for like 5 minutes.
Well, that's okay. You can go back and listen to it today.
Do we need to know where each of the things can leave and come in? Or
No. I'm just trying to give you broader perspective that the citric acid cycle is more central than it seems the way it's discussed in most other courses. Okay, there's more going on with the citric acid cycle than the impression we sometimes get based on its placement in the discussion when we often talk about it. Does that make sense? That's the idea I'm trying to get across. Okay? So, that's what the citric acid cycle looks like. When you look at, what? That's from your book, right? When you look at a metabolic pathways chart, right, the citric acid cycle is always right in the middle, right? It sort of becomes the central focal point of any metabolic pathways chart, which this is really there only to illustrate the point I was making. And so the number of things coming into the citric acid cycle, the number of things leaving the citric acid cycle, right? There's a lot going on there. That's the point of that slide. Okay? Now, the citric acid cycle starts, let's go. The citric acid cycle starts, the discussion starts. If we're continuing that theme of carbohydrate metabolism, we ended carbohydrate metabolism with pyruvate, right? Whether we took glucose through glycolysis or whether we shunted around some of the steps to do the pentose phosphate pathway, we could bring everything in, and everything ended up at pyruvate. So we're going to start with pyruvate dehydrogenase, the pyruvate dehydrogenase complex. So let's look at the pyruvate dehydrogenase complex. The pyruvate dehydrogenase complex, is this one of the ones that's on there?
I don't remember these slides.
I think there's a new slides document for the citric acid cycle. If not, let me know, because I've got these as a separate thing I can generate. Okay? So, the pyruvate dehydrogenase complex is, when we say complex, that's an understatement. The pyruvate dehydrogenase complex, let's see, it's located in the mitochondrial matrix, right? It's a mitochondrial enzyme. It looks roughly cubic in prokaryotes. It looks more dodecahedral in prokaryotes. There are at least 3 distinct enzyme activities. There's a pyruvate dehydrogenase activity, which actually acts as a pyruvate decarboxylase step. There's a dihydrolipoyl transacetylase activity, which moves the acetyl group from one place to another. And then there's the dihydrolipoyl dehydrogenase, which really, this is where the oxidation reduction occurs that creates usable energy. And it's really about returning the coenzyme of this step back to its active form. So there's a lot going on there. In a prokaryote, it can have 60 subunits. In a eukaryote, at least 96 subunits, multiple copies of each one. It's kind of big. It's got a total molecular weight in the millions, right, in the complex. So when we say an enzyme complex, this is truly an enzyme complex. You've got to look on your face like you want to ask the question.
It's just a group.
Hmm?
It's just a group of enzymes together.
It's just a group of proteins together. Under an electron microscope, it's actually visible. And so when we talk about it being, roughly cubic looking in prokaryotes, you can see the roughly cubic structure. I mean, it's a big enough protein complex that we can see it with an electron microscope. So it's big. It's built around a core where you continually build proteins on the central core. Let's see. This is from a site at UCLA showing the three-dimensional structure of the complex. So if we come back to this, right, this slide, we've got three different enzyme activities. Right? We've got an E1 enzyme activity, which is the pyruvate dehydrogenase activity. It has thiamine pyrophosphate as its coenzyme. Now, when did we encounter thiamine pyrophosphate before?
Is it DPP? Pyruvate, or the other decarboxylase?
Yeah, the pyruvate decarboxylase. And really, E1 does the same reaction that pyruvate decarboxylase did when we were talking about fermentation. Right? So it's going to decarboxylate pyruvate, but it's going to, in the end, transfer that, instead of hydrolyzing the acetyl group away as acetaldehyde, it's going to transfer that group to something else. E2 is the dihydrolipoyl transacetylase. It's got a coenzyme lipoamide, and we'll talk about that. The E3 is the dihydrolipoyl dehydrogenase, right? It's responsible for oxidizing the lipoamide at the end of this reaction to return it to its active form. And the dihydrolipoyl dehydrogenase has, as part of its structure, an FAD, right? Remember that one of the differences between FAD and NAD is that FAD tends to be involved in part of the protein tertiary structure, while NAD tends to be bound and released like a substrate, right? It tends to act more as a co-substrate than a coenzyme. So the E3 enzyme has FAD. Now, what wasn't included on the other slide is that in eukaryotes, there is an associated protein kinase and protein phosphatase that are there to help control the pyruvate dehydrogenase. And then, in terms of soluble coenzymes, it uses coenzyme A. Coenzyme A interacts with E2. And it uses NAD. NAD interacts with E3. Okay, so that's all the components of the pyruvate dehydrogenase complex.